Thanks a lot, David and Ran, for inviting me to give this seminar. It's a great honor for me. I've only briefly met Carl van Vreeswijk, but I really enjoyed meeting him, and it's great to be able to speak at a neuroscience seminar in his honor. I'll talk to you today mostly about my postdoctoral work, and then in the last part of the talk, there'll be a little bit about more recent collaborations. So since I moved to Marseille, every morning I face this very difficult choice. Drive in my car, and the campus is a bit far away. It's actually close to the Calanque, which have wonderful beaches. So every morning I encounter these road signs that tell me, turn left and go to the university and write some very meaningful equation that will change the course of science, or turn right and enjoy a relaxing day at the beach without telling my wife who's home with the kids. So in this space between the sensory inputs, the road signs, and the action that I have to take a couple of seconds or minutes later is what the great psychologist Viktor Frankl, talked about in this term. So between the stimulus, so the sensory input, and the response, the action, there is a space. And in this space or time lies our freedom and power to choose our responses. So this is what interests me and what I've devoted my research life so far. And particularly, I've concentrated into trying to understand how the brain bridges the sensory input with an action through this time that we will call delay for the purpose of the talk. And typically this delay lasts for seconds to minutes. So it's a time scale that it's much longer than the typical time scale in which a neuron can maintain information by itself. So what I will concentrate my attention on is a very simple paradigm where you have a sensory stimulus that could be a visual stimulus, like a blue arrow or a red arrow. And this stimulus is matched with an action that needs to be taken at a future time. And the stimulus disappears in between. The time in between these two stimulus and action is called delay. And this is where cognitive functions such as short-term memory and action selection needs to happen so that we will plan for the proper motor action. So if we just think about this in the most naive way, there must be a place in the brain where there is some activity, right, that should be persistent, meaning that the stimulus changes its course, and then this change lasts until the end of the delay so that the action can be performed. And it has to be selective, meaning that the two different stimuli need to give rise to two different trajectories. This doesn't have to be fine rate, right? There are many activity silent models, but somewhere in the brain there should be something like this. And history has shown that units that show this kind of activity have been found. First in some fantastic work in monkey, prefrontal cortex, in the late 80s, and then more recently also in rodents. And particularly in this task from the Brody lab, rats were asked to poke left or right depending on a sensory stimulus like the one I just showed you. And when recording from FOF, they found the presence of these neurons that show persistent and selective activity for a few seconds. So my questions during my years at Janelia and my recent start at the lab are, first, what neural mechanisms underlie these memory-guided decisions? And for this, I teamed up, as David said, with the lab of Karl Svoboda, who's now at the Allen Institute, and Hidehiko Inagaki, who was a postdoc at Janelia, but now has his own lab in Max Planck, Florida. For the second question, which is how new sensory information impact the decision during the delay, I worked with Arseny Finkelstein, who at the time was also a postdoc at Janelia in the lab of Karl, and then now has his own lab at University of Tel Aviv. And in the last part, I will show you some preliminary results on how these mechanisms are probably emerging during learning. So let's start with the simplest possible model one can think of, an extremely simple fine rate equation for one neuron. So there's a leaky term, and a term that signals the presence of the stimulus. So whenever the stimulus is arriving, right, there is an integration of the stimulus, the activity goes up. But if we just rely on a single neuron, this tau, this membrane time constant, is typically of the order of 150, 200 milliseconds maximum. So whatever integration of the stimulus has been performed will die off and will not be read out by whichever region will perform the action. So by the end of the delay, there will be no information about which stimulus had been received. So if we want to understand these kind of computations, we may want to look at networks. And so for this effort, I employed essentially two different methods. One, hypothesis-driven networks rooted in dynamical systems. And I examined several different hypotheses, right? It could be feed-forward chains, attractor networks of different kinds, even single-cell mechanisms, right? And at the same time, I've also used data-driven methods from machine learning, particularly by using recurrent neural networks to train these networks to reproduce the data and then inspect them to understand the mechanism. I will only show you two of the hypotheses that we looked at. There's more in the paper, and I'm happy to talk more in the Q&A session. But one very popular network mechanism that had been proposed is the line attractor or integrator. For this mechanism, right, we take the same equation we have before. Now we have our neuron is connected to a lot of our neurons, and the W is the connectivity matrix. If we are smart in the way we set up the W, we can see that at least for some directions in the neural space, we can now counter the leak. Whenever this happens, right, if the stimulus has some overlap with this subspace in which the leak is countered, then when the stimulus is integrated, the activity goes up, and then it will stay there for the duration of the delay, because nothing else will intervene. So if we think of this in terms of a little bit borrowed from physics, right? We can think of this as some energy landscape in which the state, the neural state lies. And at least along the direction where this canceling of the leak is happening, then we can look at a very flat stability landscape. So whenever the stimulus arrives, the activity moves and stays there because the minimum is still at the same level. So in the neural state space, as I said, this looks like a line, and that's why it's named line attractor. This also happens obviously for the opposite stimulus, and one feature, right, of the line attractors, as it's probably obvious to most of you, is that every other state in between is also stable. So you can have graded integration of stimuli. And this is a model that has been successfully applied to oculomotor integrator, for example, to explain the stability of the eye position after a saccade. A second very popular network mechanism that also had been proposed decades ago relies on discrete attractors. For discrete attractors, we take the same equation as before, but now there is a nonlinearity that is just outside, right, of the product of the connectivity matrix times the finite rate vector that contains the activity of the other neurons. And so if this nonlinearity has, for example, a sigmoidal shape, then there could be special states, right, where the activity is stable. So if we think in the same way, now this stability landscape, the energy landscape contains minima that lie at the bottom of these valleys, right, like in physics, essentially, like a double well model. And in the neural state space now, instead of having a stable line, there will be stable points surrounded by basins of attraction. So where the activity lies in one of these basins, it will be attracted to the minimum. So in the case where we have two competing actions, this will really look like a double well that most of you have seen in physics. And this is also a model that obviously has been proposed before, as I said, and has found application for auto-associative memory, so more for long-term memories, in the work of John Hopfield, Daniel Amit, and Xiaojing Wang more recently. So we wanted to ask the question, how can we distinguish between these different hypotheses in the brain? And luckily, at the time, the lab of Carl had developed a very simple task that looks exactly like the one I told you about, where sensory instruction is separated by a movement. And in between, there's this delay that lasts from 1 to 3 seconds. So mice are asked to wait before performing a movement, and then a go cue will tell them, now you can perform the movement. So one very lucky aspect of this task is that when one optogenetically inactivates each area on the cortical sheet, so the cortical level, during the delay, during the entire duration of the delay, there's only one area that shows a change in the performance of the mice when silenced. And this area is part of frontal cortex, of motor cortex, and it's called by the lab of Carl, anterior lateral motor cortex. The different name is also because of something different that this area does. And what we demonstrated, and what they demonstrated in previous work, is that this area is necessary for motor planning in these kind of tasks. So how does a typical neuron look in this area? Well, it looks like the units that I showed you before in the rat FOF. So this is an example of one recorded unit during this task. So the activity, as you see, becomes persistent once the stimulus arrives in this S period, that is the sample period. And the persistency lasts for the entire duration of the delay. Plus, the activity is selective, meaning we have cells that prefer a particular direction in the future action. This cell, for example, prefers leaking right, so it can predict a leak to the right. And we know this because when we look at error trials, the cell actually flips. So you can predict an error by looking at this kind of cell. A second lucky aspect of this task is that the activity appears to be very low dimension. So when we compute PCA or any dimensionality reduction technique, we realize that essentially with the first two modes, we can capture 95% of the variability. And these two modes capture two important aspects of the task. First mode captures the axis of maximal separation between the leak right and leak left trajectories at the end of the delay. And the second mode, this RM mode, is a ramping mode. So it's a mode that captures essentially the changes in time from the sample to the end of the delay. So we can take all our cells recorded, project them onto the first mode, the CD, the coding direction, and we get a picture that looks like this, where essentially the blue trajectory is for leak right trials, the red is for leak left. I'm showing you recordings from left ALM, and that's why the picture is not symmetric. If you take both the ALMs, the picture becomes fully symmetric. So how can these models capture a dynamics that doesn't really look like the one I showed you before, that it's just an integration and then flat, but it actually looks like it's slowly ramping. So it's rolling towards a higher activity state. Well, for the integrator, if we just rely on this intuition with the energy landscape, we can just tilt it just a little bit, right? So that it's still pretty slow, the motion along this axis, but it still moves to a different level. For the discrete attractor, it's a little more complicated because you can't really move basins around. But I'll ask you to trust me that we can do this essentially by adding an external stimulus that I will talk about later. So how do we distinguish between the models? Well, the first thing one can think of is to perturb the activity around the activity state. And one thing we can do now, it's optogenetic silencing, but for a duration that is much smaller than the duration of the delay. So if we inactivate for 500-600 milliseconds, then we expect the models essentially to have very different behavior. For the integrator, silencing will essentially move the activity back to an activity of almost zero, which means that the activity will go back and then restart the rolling down. The picture is different for the attractor, since when we now push the activity back, there is a barrier, and this barrier is pretty steep. So what happens, right, is that there is a recall mechanism, and the activity will be called back, and will continue the trajectory that should have had in absence of the inactivation. So we can look at the data, because the eco did exactly this experiment. So during the inactivation, you see the trajectories approach 0, meaning the firing rate is really low. And then once we release the optogenetic inactivation, the activity flows back to the original trajectory. It doesn't restart. It actually goes up pretty quickly. So we think that this favors a discrete attractor picture. We can then look at what happens when the photoinhibition causes an error, right? In this case, we can take trials that were not errors before the photoinhibition, and we can look at the effect of the photoinhibition when the mouse ends up leaking in the incorrect side. And what we see is that now there is a complete flip, right, with the red trajectory approaching the blue and the blue approaching the red, which seems to suggest that we're able now, with a strong silencing, to cause the activity to jump the barrier and end up in the other attractor basin. So all this can be captured by a rather simple model where we have essentially two excitatory populations that encode the sensory input and transform it into neural activity, right, that will be read out to perform an action. And it's sufficient essentially to link these two populations via a shared inhibition that plays the role of a selector, so that this network in the end behaves like a winner-take-all. As I said before, though, if we want to also capture the slow ramping, we have to introduce a ramping from the outside. And this is to help the network create a time scale, right, that is slow enough. One prediction, right, from this hypothesis is that the ramping input is not only changing the level of the activity, but it's also changing the shape of the energy landscape. So if we take, for example, an early time during the delay, the ramping input is not very strong. The two basins are close to each other and rather shallow. If we look at the late time during the delay, now the ramping input is much higher. It excites more the two excited populations, and this creates much deeper basins separated by a higher barrier and more distant. So one thing that we thought about when we were thinking about this is that these mice are always trained with the same fixed delays. So a mouse sees the same 2 seconds delay for hundreds of trials. So what happens if instead mice are now trained with a random distribution of trials where they cannot really find any mean delay? Well, the prediction from the model is that this ramping input that now creates the slow timing ramping will switch back, so that now mice are ready for the shortest possible delay so that they don't miss the reward. And that's exactly what happens. So if we train mice on a random delay, we don't see anymore the slow ramp. The activity of the recorded cells in ALM now switches to the higher level very quickly and stays there. And this is true also at the level of the population. We performed, IDECO performed the silencing experiment in this case too, and essentially confirmed the same picture we had before. Okay, so we hypothesize now that the dynamics during the delay are explained by a double well attractor. But what happens if we're able not only to perform this very rough silencing, but if we can essentially introduce more sensory informations during the delay and see essentially how the dynamics are changing? Well, one hypothesis could be that the activity just follows essentially the same dynamics that happened during the silencing. But it could also be that the integration happens in a completely different way with a different mechanism. And so the activity could, for example, end up at some level that has nothing to do with the original trajectories, or be called back. So for this experiment, teamed up with Arseni, who at the time had developed a similar task with tiny differences. Essentially, the behavioral output is the same. There is a lick left and a lick right. But the sensory stimulation is directly given to sensory cortex, to one barrel in sensory cortex, in barrel cortex. And so mice are trained now to detect the stimulation. In that case, they will have to lick right if they want to receive the reward, or lick left whenever they don't detect the presence of a stimulation. And this allows for a much better control of, for example, the error rate, because Arseni can tilt the amplitude and the duration of the stimulus. So we essentially had 4 types of trials, one in which the stimulus is presented during the sample, and then nothing happens during the delay, one in which no stimulus is presented, and 2 distractor trials, one in which the stimulus is presented early in the delay, and one in which is presented late. And what's interesting is that the behavior already shows us that mice are essentially committing a lot of mistakes. They're fooled by the early distractor stimulus, but they're not at all by the late distractor. So the late distractor will not cause many errors. So this also can be explained, right, by a similar model to the one I showed you. And in fact, the prediction I showed you is compatible with what I just showed you. But I wanted to make use of the great variety of neurons that Arseni was recording. So we have cells, these are just 4 examples, that don't behave like the typical cells that you have in these simple EI networks. For example, they can encode the stimulus and then decay back quite quickly. They can show typical behavior, or they can even switch selectivity. So they can start being more selective for licking left, and then they end up by being more selective by licking right to the licking right during the delay. Also, if you look at the distribution of fine rates during delay epoch, they typically don't look at all like the distributions that you have in these kind of models. The gray distribution is the true distribution of recorded neurons that resembles a log normal distribution, and not the peaked Gaussian distribution that we typically put in this model. So the idea here was to use the power of machine learning. And for this, I developed a recurrent neural network, very simple, in which the equation it's very similar to the one I just showed you, in which, though, we're training the recurrent connections so that each unit will look like a neuron recorded by Arseni. So there is a cost function, a global cost function that is the sum of the cost function for each unit. So at the end of training, we need all units to look exactly like the neurons. And the inputs are given through weights that are random. So these projection weights are chosen at random for each train. And I can train the network with or without the ramping to check whether the network is able to create this slow timescale by itself, or it needs a little help to do that. And I forgot to mention the nonlinearity is a sigmoidal nonlinearity. So it turns out that the network can learn, and then we want to test the network on a different set of trials. And what I thought of doing is to test it on the trials that look like the Distractor trials that I showed you. So trials in which there is an extra stimulus appearing either early or late during the delay. So the network has never seen trials like this. We're testing it. What was quite surprising when I looked at this was that the behavior looks very similar to mouse behavior, meaning that the early distractor trial types cause many mistakes in the network too, but it doesn't happen in the late distractor. The trajectories look essentially like a cleaned up version of the recorded trajectories, considering that in ALM, I'm showing you one session because there are some differences essentially in the number of selective neurons, while this is the all sessions collapsed. And one super interesting thing about these networks that David Susilo and Omri Barak have shown now more than 10 years ago is that they're very accessible. They're not complicated black boxes. like, for example, other more complex dynamical system models that are paired up with Bayesian inference. With these networks, you can take the dynamics, you can inspect it, for example, by linearizing or by testing it with random perturbations, and you can check for interesting manifolds, interesting fixed points, etc. So what this network that I trained ended up doing is the same picture that I showed you for the simple dynamical system. So it implements low-dimensional dynamics, and along this one dimension, essentially, in which the computation takes place, there is a bifurcation. So from the baseline state before the ramping input starts, there is only one stable state. When the ramping input is fed in, there is a bifurcation of which the ramping input is the parameter. There are two Bayesians now appearing, and these two Bayesians behave in the same way I showed you before. So one prediction, right, extra prediction, I would say, from this network is that this external ramp controls the distance and the depth of the Bayesians, not only during a trial, but also across trials. It could be, for example, that mice are, at the end of the session, a bit more demotivated. In that case, they're maybe a little less able to predict timing, for example. So this external ramp could be weaker in some trials. In that case, the prediction is the Bayesians are going to be shallower. So if we now go back to the data, look for the ramping mode, which was the second mode that I showed you in the beginning, right? And now we bin trials based on the slope of the ramping mode. So on how strong in the data the ramping mode is. And what we found is that if we do that, the probability that the distractor will cause a mistake is much higher if the ramping mode is weak, as a small slope, which is what the network had predicted. So we think this is pretty good evidence that there is some kind of controlling signal that we hypothesize predicts time, but could also encode motivation. And this signal is changing the shape of the computation in time, within a trial and across trials. So just to summarize this part, activity in frontal cortex, we find it to be compatible with discrete attractor dynamics, at least during the DA epoch. The ramping input controls the separation between these two attractor Bayesians and their depth. This dynamics that it's dynamical, this attractor dynamics that is dynamical because of the ramping input, can also control the way sensory information flows from sensory cortex to ALM and can provide more or less robustness in time. As a consequence, because these slow dynamics are a consequence of temporal predictions or motivations, they're not necessary for memory maintenance, meaning that you can train mice on a random distribution of delays. This slow dynamics disappear from the delay. You have this very quick switch, but the memory is still there. And something I haven't shown you, but that IDEICO tested experimentally, is that single cell dynamics does not seem to be very important for at least in this task and in mice. So if you, for example, inject currents in single cells at different times during the trial and measure, for example, the decay time of these perturbations, we don't see any major differences. So we don't see any nonlinear changes in single cells. It still may be that at the level of the dendrites, this is true. At the level of the soma, we don't see it. So more recently, we started thinking about how these slow dynamics appear and emerge during learning. And for this, keep collaborating with IDEICO in Florida, who has developed an even simpler task now. The task now is not a binary task, is simply a task in which mice need to lick after a certain time when they heard a sound that just signals the onset of the trial. So if they happen to lick late enough, they will receive a reward. If they lick too early, there is no reward. And the length of the delay is not explicitly specified to mice. So it turns out that if you train mice to do this progressively, so by progressively increasing the duration of the delay, mice can learn to wait up to 2 seconds, or longer, actually, not up to 2 seconds, just in this case, it's 2 seconds for this particular mouse. And what's very cool from the lab of IDEICO is that they can now record activity at the different stages of learning. So from the first session to the last. And you can see here, the blue is the first session. So when mice were asked to wait only 200 milliseconds, or less actually, 150. And the red is towards the end of the session when mice are asked to wait up to 1 second. And so you can essentially notice two tendencies. First, that the cue response, or the first integration, becomes much smaller with time. And second, that the slow ramping activity seems to be emerging, which is what we expect based on the previous trials. So we can time warp these traces to check, for example, whether the final state is more or less similar, and we do find it to be quite similar. In fact, this difference is not significant. And one cool thing IDEICO could do is to block essentially plasticity in a lamp during learning. And he did this by blocking CAMK2, which is a promoter of plasticity. And what we see is if they do that in ALM, we do see that mice fail to learn. So they cannot learn to wait. If we do that, for example, in motor cortex, this doesn't happen. So ALM seems to be the locus where the plasticity is important also for the emergence of these dynamics. We also have now access to the set-specific contribution. I don't want to spend too much time into this, but we know already that there are two important classes of cells, pyramidal tract cells and intrateloencephalic cells. These pyramidal tract cells, the PT cells, are found to be the ones that affect learning. So if you block CAMK2 in PT cells, postsynaptic to PT cells, then you see essentially the mice cannot learn. So we already have a little bit of an idea of how this circuit should look like. And we know that ALM is recurrently connected to thalamic neurons. But to try to explain this with a model that captures the same aspects of the ones I showed you before, I built a very simple model with thalamus and ALM, in which ALM has essentially 3 cell types, these IT cells that are important for maintaining the dynamics, but are not important for the emergence of the dynamics during learning. Inhibitory cells that obviously are maintaining balance, and PT cells that we know are important, linked with thalamus. So this is very simple model with essentially 4 variables. And all these cells, or in different fashions, can receive the cue that signals the beginning of the trial. So to capture learning, what I did was essentially introduce fluctuations in the cue. Whenever these fluctuations are strong enough, because of the way the circuit is set up, there can be a slow dynamics that it's a tiny bit slower. This slower dynamics triggers a reward because the mouse was able to wait, for example, more than 100 milliseconds. And this reward, it's linked to a change in the plasticity. And for example, we obviously tested more than one model, but one connection that could change is the connection from sensory cortex to the PT cells. So if we do this with the model, and here I'm showing you the version where the changing connection is the one from the cue to PT cells, the model can learn like mice do, simply as a consequence of fluctuations in the cue input, and then some reinforcement rule on the weights. But by the end of the learning and during the learning, we can follow how the model performs. And this is the activity of the model. The model is very simple. It's a linear threshold model, so it doesn't have any fancy dynamics, fancy tuning. But we can see that essentially we capture the effect of the smaller cue and the emergence of the slow ramping dynamics. if we then inspect the model, which is obviously a bit trivial because I set it up like this, we can see it. Essentially, what's happening is that the response of the cue is getting smaller because of the potentiation, which means essentially that the cue is sending the activity on a slow manifold at a different position. So in early leak trials, the cue is able to push the activity further down this slow axis. And in late leak trials, this is not true. It puts the activity at a higher, further away state. And that's why it takes longer for the slow dynamics to develop. So it's an extremely simple mechanism, but it seems to explain quite well what's happening. And what's cool is that we can test the ability of the model to explain the behavior in expert animals. So when animals, after 3,000 trials, they become quite good at switching between different delay durations. So at that point, there is no more progressive increase. There are blocks of trials in which mice are asked to wait 1 second or 3 seconds, and then vice versa. So during these blocks, right, the activity can be recorded. Here I'm showing you blocks between 1 and 2 seconds. And you can see that now the slow ramping activity is already there. There's no more changes in the cue. The activity simply scales with the demand, so with the duration of the delay. And if we warp them, this is even more clear. So we want to use the same model, the one that has learned with this very simple correlation-based rule, to try to capture this behavior too. So to do this, what we can do is introduce an extra input that is a tonic input, that it's essentially there for the entire duration of the block. So this input signals just in which block, so in which duration of the delay the mouse expects to be. And just by introducing this input, we can scale the activity quite effectively. And so now we can essentially have, with a lower input, a slower convergence to the fixed point, which lies at the end of the slow manifold. And with a stronger input, these dynamics happens faster, which is probably quite intuitive. Now, in this case, right, what's happening, if we look at the energy landscape, is that the tonic input is actually changing the landscape itself, and it's making the slow manifold slower. So I'm not showing you everything because also I'm about to be out of time, but just to conclude on this part, we show that these discrete attractors with a slow ramping dynamics can emerge from a simple correlation-based learning rule. That the same network that has been developed for that task can generalize to explain expert behavior just by introducing a tonic input. That, by the way, we can also identify in neural data, and this will be subject of a publication. We have already a bioarchival. And I didn't show you, but we can achieve the same effect with long-term depression instead of potentiation. So we can set up the circuit in a different state, and instead of having potentiation of this connections, we can use depression and obtain a very similar result. So now what we plan to do is essentially to use the predictions for the model to develop experiments that can make us distinguish between these different mechanisms. Let me thank all the people who greatly contributed to this work. First, my supervisor at Janilia, Sandro Romani, who has been a great mentor all these years. Obviously, Carl Svoboda, who has shared all this data, Arseni and Ideiko, who made all these wonderful experiments. I, as David said at the beginning, started my lab recently in Marseille. I already have for first 2 members, but a bit of shameful advertising. I would love to, get in touch with any of you if you're looking for a job, because I'm hiring postdocs and PhD students. And thanks a lot again.